Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture number 54, where under the broad group of phenolics, I am going to cover biosynthesis of anthocyanins and metabolic pathway engineering for enhanced anthocyanin production. So, let us go to the concept to be covered. So, we will talk about the biosynthesis of anthocyanins, and then we will discuss the issue of anthocyanidin and anthocyanin. Next, we will talk about the structural diversity of anthocyanins, and then finally, we will see how anthocyanins are, once they produced in the cytosol, how they are transported into the vacuoles. Okay. So, let us go to this previous slide. This slide I have shown several times, but this is the basic one, basically the overview of phenylpropanoid metabolism. So, the anthocyanin is basically coming under broadly the flavonoids. So, as you know, flavonoids are C6, C3, C6 compounds, and anthocyanins are just one of these. So, and this basically originates from the phenylpropanoid pathway by joining malonyl CoA with the paracomadoyl CoA, leading to the formation of chalcone. So, the chalcone synthase is the first enzyme. So, I have spoken about chalcone synthase several times. So, this is also no exception. Chalcone synthase is a starting point. Core pathway for anthocyanin biosynthesis, and basically showing the relationship between flavonol and flavone. So, these aspect I have covered in little bit detail, particularly on the flavone and flavonols in the previous class. So, let me make a brief overview about this, that as I mentioned just now, phenylpropanoid start way originating with phenylalanine converting into cinnamic acid, then cinnamate 4 hydroxylase, then 4 Cl, which makes the activated hydroxycinnamate, which is paracomadoyl CoA, because the CoA molecule is joined there. So, that is more active, and that joins with 3 molecules of malonyl CoA and makes the chalcone, or which is also called naringenin chalcone, because it next step it makes through isomerization, it makes naringenin. Okay. So, the enzymes responsible for this is chalcone synthase, and next is chalcone isomerase. So, the 3 molecular malonyl CoA joins with paracomadoyl CoA and forms the chalcone. So, that is called Clausen condensation, whereas for stilbene it is called aldol condensation. Stilbene synthesis also requires 3 molecular malonyl CoA, but the mechanism of condensation was different than that of this one. So, once naringenin is produced, then a part of naringenin may be utilized for the production of flavones, flavone synthase, and then but the main pool of naringenin may further converted into dihydroflavonol by the action of F3H. F3H is basically flavanone 3 hydroxylase, then F3 prime H, which is flavonoid 3 prime hydroxylase, and F3 prime 5 prime H, which is flavonoid 3 prime 5 prime hydroxylase. We will see this in due course, and that makes dihydroflavonol, and dihydroflavonol one route may produce flavonol, whereas the route which is meant to produce anthocyanin in specific plants. So, that pathway moves in this direction, where dihydroflavonol reductase will come and then followed by anthocyanin synthase. Okay. So, it is basically anthocyanidin synthase, and then once anthocyanidin is formed, then there will be a sugar molecule addition that leads to the formation of anthocyanin. So, the anthocyanidin and anthocyanin differs is that first comes to anthocyanidin, which is basically a aglycone, whereas when anthocyanidin joins with sugar, then it makes anthocyanin, and then it is basically stored in the vacuole. So, this is from the structural point of view. As I said that chalcone synthase makes chalcone, which is tetrahydroxy chalcone. So, this is the structure of tetrahydroxy chalcone, and so do not see CHR here, CHR has no relevance particularly here. So, you can simply cut it. STS also we are not discussing here, but what we will be discussing the next step is this is converted into by this chalcone isomerase forming naringenin. So, the naringenin is basically coming under flavanone. So, naringenin is flavanone. So, naringenin can be converted into iriodictyol, okay, and the pathway may utilize in producing dihydroquercetin, whereas dihydroquercetin can also be produced from dihydrocampherol directly through this route as well. Okay. And similarly, what is showing here that pentahydroxyflavanone can also produced and then F3H, but this is also possible. So, here if you see the difference between these three structures. So, here particularly what you see here is this position OH is there. So, this is 4 position OH is there, but here there are additional OH here in addition to this one. So, 2 OH. So, one hydroxylation happens. So, here the enzyme is called F3 prime H, that means flavonoid 3 prime hydroxylase. Now, if you compare this one with this one, so here there are 2 more. So, this is called F3 prime 5 prime H, flavonoid 3 prime 5 prime hydroxylase, which makes dihydromyricetin. And then these 3 pathways contribute to different anthocyanin structures. For example, the one cyanidin, which is basically magenta towards red color. So, the cyanidin is this one. Again, the common enzyme is DFR, but these DFR are specific. So, one DFR which is specific for dihydroquercetin will not work for dihydrocampherol or dihydromyricetin. So, these are specific, but the generic name is DFR, dihydroflavonol reductase, because all these are dihydroflavonols. So, all these 3 are basically dihydroflavonols, plural I want. Okay. So, that is why dihydroflavonol reductase is there. So, which converts dihydroflavonol into leucoanthocyanidin. So, so that means that is colorless. So, leucocyanidin, leuco means colorless, leucopelargonidin or leucodelphinidin. And the next step is this by the action of anthocyanidin synthase, which converts leucocyanidin to cyanidin or leucopelargonidin to pelargonidin. So, pelargonidin are generally yellow or orange in color, and the third one is this, the delphinidin. Delphinidin are blue in color. So, delphinidin requires 2 hydroxylation to happen, that is F3 prime 5 prime H, whereas cyanidin is only 1. Whereas for pelargonidin no substitution is required, only this position H will remain. So, this is in brief about the biosynthesis of anthocyanins. Okay. And what I have said in the previous class, that these dihydroquercetin or dihydrocamphorol or dihydromyricetin can be converted into the either to myricetin, which is flavonol. So, that is called flavonol synthase, which I have shown. So, in the previous class. So, for example, this one by the action of flavonol synthase FLS will be converted into, this is L capital. Will be converted into dihydroquercetin will be converted into quercetin. So, quercetin means if we draw the structure, it will be like this. So, this structure is dihydromyricetin. So, the when it will become myricetin, there will be only one bond formation here. So, sorry. So, this becomes quercetin or myricetin, sorry. This is myricetin, which is basically a flavonol. So, that is the side pathway, but main pathway moves in these three direction. So, let us now move into the next slide. So, pelargonidine is the basic structure, as you see that in these two positions are open. So, no hydroxylation added yet, but cyanidine, what I have shown that this position 1 hydroxyl is added, and in case of delphinidine 2 hydroxyl is added. So, pelargonidine is responsible for yellow orange color. So, here there is a picture of geranium, which is pelargonium. There are multiple species, and the cyanidine is a common example is rose, and delphinidine you find it the plant which is commonly grown in this winter season, which is called larkspur, which is the English name, but the scientific name is delphinium. So, that produces blue flowers. Okay. So, now what we will see how this structural changes occur and how these different anthocyanins are created. So, now let us go to the whiteboard. So, here what we will see first is we will draw the pelargonidine once again. So, this is the structure of pelargonidine. So, which is a anthocyanidine. Now, if here if this OH is replaced here, the hydrogen is replaced and sugar is added, then basically it forms pelargonine, sometimes they use this term, which is anthocyanin. Now, with this structure, so what we see now, this structure is still not correct because I have not added OH here, and two more things to be telling here, that this will be the 3 prime position and this will be the 5 prime position. So, now let us see the anthocyanidins. Name of antho then we will put substituents and then we will put the color. I use the British spelling color, COLOUR. So, first one is pelargonidin. Pelargonidin. Substituents at 3 prime and 5 prime, as such there is not, it is only H, and what we see as a result, the color is orange or yellow or red, sometimes they call. So, orange red. Next one is cyanidin. Cyanidin 3 prime OH and the color will be red. Peonidin three prime will be now OCH3, color will be pink, red. Next is delphinidin. Here 3 prime OH and 5 prime will also be OH, and this will be color will be bluish purple. Next is petunidin. Petunidin 3 prime will be OCH3 and 5 prime will be OH. The color will be purple, and then malvidin, MALVIDIN 3 prime OCH3, 5 prime also OCH3, and the color will be reddish purple. So, you see that these substituents at these positions, that is at 3 prime and at 5 prime, really makes difference in the color of the anthocyanin. So, one thing to be noticed that, as I have said, that cyanidin formation requires from dihydrocampherol, cyanidin formation requires F3 prime H, that is flavonoid 3 prime hydroxylase, which adds the at 3 prime position hydroxy group. Okay. So, that is why this is 3 prime OH, and then next what happens, the pionidine, that 3 prime position hydroxyl will be replaced by methoxy, a methyl donor will be there. So, that converted into pionidine. So, that means pionidine cannot be formed without the formation of cyanidin. First cyanidin is formed, then it will be converted into pionidine. So, because I have said that is a thumb rule, first there will be hydroxylation, then followed by methoxy. Similarly, for delphinidine, what I said is basically it is F 3 prime 5 prime H. So, dihydrocampherol converted into dihydroquercetin, dihydromyricetin by this F 3 prime 5 prime H. So, that is why the 3 prime and 5 prime OH is added and delphinidine is produced. Then in the next step what happens, the 3 prime OH will be replaced by OCH3, it converted into pitunidine. So, that means from delphinidine pitunidine will be produced, and finally, from pitunidine malvidine is produced. As you see from here, this OH now become OCH3. Okay. Similarly, like here also this OH now become OCH3, like this way. So, it follows the straightforward chemistry. Okay. Now, what I have discussed so far is this, that we have discussed chalcone synthase, chalcone isomerase, flavonone 3 hydroxylase, which converts naringenin to dihydrocampherol. So, these are all dihydroflavonols. So, these are dihydroflavonols. So, naringenins are flavanones, and this step is chalcones. Okay. And in the next step, what we see, that this will be converted into dihydrocampherol either by F 3 prime H will be converted into dihydroquercetin, and which eventually makes the formation of cyanidin. But cyanidin, what happens, actually this is shown in in one single step. Actually, this is 3 different steps are involved. So, first one is DFR. So, DFR converts dihydroquercetin to leucoanthocyanidin, which I have shown in the previous slide, and then leucoanthocyanidin by anthocyanidin synthase, AS or ANS is same, ASN or ANS is same, which makes the cyanidin. So, that is still aglycone, and then GT stands for glucosyl transferase. Why 3 GT? Because at 3 position this will be added. So, this will be glucosyl transferase, and then it makes cyanidin 3 glucoside. So, what you see here, this cyanidin 3 glucoside. So, this cyanidin 3 glucoside subsequently converted into pionidine, as I have shown you. So, this is becoming OCH3 here, then this is OH, and then what happens, that different sugar molecule can join and it makes like so I will tell you the different sugars involved in this decorating the anthocyanin in some of the in later slide. Okay. And the delphinidine purple. Okay. Similarly, these are the finally these are the anthocyanidin, and then this basically these are finally the anthocyanins. because the sugar is attached to here. So, this is a brief overview, already I have discussed. Now, coming to the decoration. So, this is the B ring, which is important. So, here actually these positions changes occur, okay. And this basically B ring is coming from the phenyl propanoid group, and the A ring is basically coming from the malonyl. So, what we see that there is a hydroxylation. So, hydroxylation pelargonidine, as this is 4 fine. Cyanidine, what we see, addition 1 more, in delphinidine what we see 2 more. So, this is all hydroxylation. Then methylation. So, this one undergoes first this one undergoes methylation, cyanidine in case of okay. So, it makes pionidine, this is a methyl group added, and in case of this one there is methylation here as well, which forms pitunidine. In malvidine there are 2 methylation. So, like this way. And not only that, what are the sugar glycosyl units added? One is arabinose can be added, galactose can be added, glucose is common, rhamnose is also common, xylose can be added, and rutinose can be added. So, similarly, the rutinosyl transferase, rhamnosyl transferase, glucosyl transferase, all these enzymes are functioning, and that leads to different patterns of anthocyanins with variation in the color. Now, apart from this, common acyl units can be joined with the anthocyanin, like acetic acid can be joined, caffeic acid can be joined, which makes complex anthocyanins. Even comaric acid, ferulic acid, malonic acid can be joined, or even gallic acid, synapic acid, succinic acid can also be joined, and leading to the formation of complex anthocyanins. For example, pitunidine 3 paracumaryl rutinoside 5 glucoside. You see how big is the molecule. So many at least you see there are 3 sugar components attached here, 1, 2, 3. Okay. So, similarly, gentiodelphine, which is delphinidine 3,5 dicaffeoyl glucoside 3 glucoside. So, maybe this is 1 caffeoyl unit coming, this is 1 caffeoyl, because caffeoyl contains 3,4 dihydroxy. So, this is caffeoyl 1, this is caffeoyl 2, and this 5,3 means that the 2 position, either 3 position, 5 position, like that way, and then it makes the things. So, this is how the anthocyanins in the natures are decorated, leading to the formation of complex anthocyanin. Now, another important point is that anthocyanins are basically stable at acidic pH. So, if the pH changes, there will be ionization effect. So, normally the pH 1 or 2 is the most stable pH for anthocyanin, and which is red in color, and at that it forms the flavinium cation. Then what happens that if you add alkali, so then at pH 4, 5 it is colorless, and next what happened at pH 6 to 7 it converted into quinodal base, and that leads to basically blue color. Okay. So, point here to be noted that and then if it is beyond 7, then it will turn into yellow, and once it turned yellow, if you change the make acidic, it will not convert back. So, that is some experiments people have done in vitro, and based on these results they worked out the complex chemistry. So, so that means that anthocyanin is very much pH sensitive. So, pH is very important for its stability. So, sometimes you see the slight changes in the anthocyanin coloration in the flower petals. So, it is not that bluish means it is all not that delphinidine. It may be possible that it is a cyanidine slightly present in alkaline or less acidic condition. In that time the red the intensity of red will be dispersed and leading to formation of bluish tint. So, that is also possible. And this is again a much more complex one, I will leave it. So, this is overview, I have already discussed this structure. So, the last part of this lecture is this: once these anthocyanins are produced, so these anthocyanins are in the cytosol, okay, methyl transferase, glucosyl transferase, then ribnosyl transferase, whatever. So, these anthocyanins cannot stay in the cytosol. So, they must be stored in the vacuole. So, how they move into the vacuole? They move, there are two hypotheses, one is through the vesicle mediated transportation. So, this is vesicle, in the next slide I will show that, or they are entering into the ER, and there they basically their GST is attached to this glutathione S transferase, which plays important role in the transportation of anthocyanin into the vacuole by through several transporters. So, two things happen, that one, either it moves through a vesicle where GST is mediating that, or without the involvement of vesicle, the anthocyanins can directly move inside the vacuole. So, and here the MAT transporter plays role, and also ABC transporter plays role in this, and here the pH, the protonation is important, because the acidic condition of the vacuole to be maintained, so that the anthocyanin remains stable. So, the vacuolar pH is very important. One important point is that if the vacuolar pH changes, anthocyanin may become unstable or may changes its structural conformation. So, this is one, and this is another way of showing that first one is that through involvement of GST, and then GST helps to transport the anthocyanin into the vacuole by the use of either ABC transporter or a MAT transporter, or anthocyanins are basically are enclosed in some vesicular structure called autophagosome, which finally fuse with the vacuole or membrane, and that then it enters, and finally the autophagosome get dispersed and anthocyanins stored. But once anthocyanins are entered inside the vacuole, then it sometimes anthocyanin vacuolar inclusions it stays. So, it joins with either a chemical molecule or a protein molecule, and it is stored there. So, tonoplast means the vacuole, vacuolar membrane, okay. And the so here what I am showing here is that this is simply a light microscopic view. So, here you see that this is a one thing, okay, how to show. So, this is one single cell, right, and this is single cell, what you see, this is the main vacuole portion, and here what you see, there is another this structure. So, this is basically anthocyanic vacuolar inclusions inside the tonoplast. So, these are the vacuolar inclusions, and that is inside the tonoplast, like this one. So, it is possible to see under the high resolution, and then in addition to that there are other bodies such as these things, which are basically present in the periphery of the vacuole. So, this could be possibly the vesicular structures. So, this was observed in our own laboratory under light microscope. And so, before I end this course, I will briefly tell about one metabolic engineering application, but this is not with the flower. So, flower metabolic engineering we will talk in the next class. So, what here they have done, look, the flavonoid pathway is universal, it present in every plant. So, what needs to be done, that why some plant leaves are not producing anthocyanin, because it is very temporally and spatially regulated. Now, this regulation is basically controlled by transcription factors. So, scientists what they have done, they hired two such transcription factors from Arabidopsis, which are known to enhance the anthocyanin, and that transcription factors are del and rose. So, these transcription factors are expressed in tobacco, and as a result of that what will happen, these transcription factors normally influence chalcone isomerase, chalcone synthase, you see flavonone 3 hydroxylase, dihydroflavonol reductase, and glucosyl transferase and so on. So, these transcription factors plays important role. What happens, maybe in tobacco flower anthocyanin are synthesized, but in the leaf they are not synthesized. Maybe in the leaf tissue these genes remains in the dormant condition, because that is slightly regulated by another set of some negative control was there. So, now that negative control is removed by overexpressing the transcription factors from Antirrhinum majus, not Arabidopsis, please excuse me. AM stands for Antirrhinum majus, snapdragon, and as a result of this expression what happens, the tobacco leaf, once it accumulated anthocyanin, it basically turn into blackish, which is basically more cyanidin accumulation, and from these leaf basically they have developed the cell culture, and these cell cultures produces anthocyanin in huge amount, and from which they are able to purify and isolate in the form of powder. So, this was this paper published in Metabolic Engineering 2018, and so this is the normal so this is where the AM del only expressed, and this is AM ROS 1. When both are expressed, you see the changes in the leaf color, and this is basically the cross section of the leaf. These are the cross section of the leaves. Here it is very clear that the anthocyanins are producing in huge amount, as compared here little bit, is you see from the coloration here, slide later, but here is huge. And from there basically they have developed the cell suspension culture. So, the first one in the left is basically the cell culture without anthocyanin induction, and this is basically cell culture with anthocyanin induction. Too much anthocyanin turns this cell apparently towards blackish, but it is not black, it is a too much concentrated red coloration, and this is the suspended cells which can be checked under the light microscope, and this is a comparison they have done. So, what we see here, that with AM del and AM rose and with another genes F3 prime 5 prime H. So, when F3 prime 5 prime H is not there, the anthocyanin still produce in huge amount, but when F3 prime 5 prime H is there, that they had from PH means Petunia hybrida, the concentration of anthocyanin, you see the intensity of coloration is much more, even it is much more, even furthermore probably when they have used acyl transferase, and they compared this with Vitis vinifera. Vitis vinifera is known to produce anthocyanin. Cell cultures of Vitis vinifera with elicitation can produce anthocyanin of this level, but with tobacco expressing these transcription factor genes along with the genes of the pathway, it can produce huge amount of anthocyanins, and these anthocyanins they have purified and used in different food products as well. So, this is very interesting. With this I end this class. Thank you.